Hello students, welcome back. In today's lecture, we will discuss how various cells are recognized by the immune system using the MHC protein. These cells will include normal self or host cells, transplanted cells, infected cells, and cancer cells. Shown here, we have a normal self cell or a normal host cell. Within the cell, we have a nucleus with its DNA. In order for a cell to survive and perform its function, it must always produce proteins. Shown here are representative proteins. When any cell makes proteins, some of those proteins are set aside and used by the immune system to determine the internal state of that cell. These proteins are chopped into fragments, placed onto special molecules, and presented to the immune cells on the cell surface. Let's first represent these fragments. And now, let's represent the special molecules which present the protein fragments. Let's redraw this cell so that we can add the protein fragments onto the presentation molecules. Now we will add the protein fragments to the presenting molecules. Let's redraw this cell again so that we can place the presenting molecules, now holding the protein fragments, onto the surface of the cell. Let's place these presenting molecules on the cell surface. To make things easier to visualize, I will make one of the presentation molecules larger. There's a special type of immune cell which reconnoiters and surveils the entire body, checking these fragments. The immune system then checks for protein fragments that are not part of the normal body. The top feature on the immune cell senses the protein fragment. The bottom feature on the immune cell senses the presenting molecule. This is how the interaction would take place here. If it finds abnormal proteins, it destroys that cell. This is an extremely important concept to understand. Every cell in the body that produces proteins is constantly presenting fragments of those proteins to the surface for the immune system. In essence, this mechanism allows the immune system to, quote, see inside the cell to make sure that nothing is there that should not be. Let's next see what happens with transplanted tissue. Imagine that the recipient receives a tissue transplant from a close relative, say a sibling. Let's represent the cell of a sibling. Represented here, the cell of the sibling is very similar to the recipient. Only one protein fragment is different. Let's redraw this cell with the protein fragments already on the presentation molecules. As we can see, only one presentation molecule is different. Let's redraw this cell and place these presentation molecules on the surface. As before, the immune system will check all these fragments to make sure that none is abnormal. However, in this case, it will find a protein fragment that is not part of the body. In this situation, the immune cell will inject toxins into the cell to kill it, and this tissue transplant will be rejected. In order to prevent this, the healthcare team would administer immunosuppressant drugs to weaken the immune system until the body accepts the new tissue. Let's next see what happens with infected cells and cancer cells. Because I don't have a lot of room, I will represent all these situations in the same cell. Just remember that they do occur separately. The cell here represents our infected cell. Here we have a cell that has been infected with a virus, or that has been infected with bacteria, or that is mutated to become a cancer cell. Here we have a virus which has produced viral proteins, which have been chopped up to produce viral protein fragments. Here we have a bacteria which has produced bacterial proteins, which have been chopped up to produce bacterial protein fragments. And here we have a mutated cancer gene, which has produced cancer proteins, which have been chopped up to produce cancer protein fragments. Let's redraw this cell with the protein fragments already added to the presentation molecules. Here, in addition to the normal protein fragments, we see that the abnormal protein fragments have also been added to the presentation molecule. Let's now represent these molecules on the surface of the cell. Let's also represent our immune cell. Here we have our special immune cell. When the special immune cell binds to the first fragment, it will say to itself, this is a viral protein fragment. This cell must be infected with a virus. I must kill it. When the immune cell binds to the second fragment, it will say to itself, this is a bacterial protein fragment. This cell must be infected with bacteria. I must kill it. And when the special immune cell binds to the third fragment, it will say to itself, this is a cancer protein fragment. This cell must have mutated to become a cancer cell. I must kill it. When the special immune system sees any of these abnormal fragments being presented on the cell surface, it will inject chemicals into the cell to destroy it. So these are all the scenarios for the recognition of normal cells by the immune system. Let's annotate some things. The first situation here is just recognition of a normal self cell. In the first cell, we have a nucleus, DNA, protein, and protein fragments. Here we have our nucleus, our DNA, our protein, and our protein fragments. Here on the last cell, we have a presenting molecule. The presenting molecule is also known as the major histocompatibility complex type 1 or class 1. Major histocompatibility complex type 1 or MHC1. Shown here is the immune cell with two distinct structures. The one on the top senses the protein fragment, and the one on the bottom senses the presenting molecule or the MHC1. Here we have our immune cell with the protein fragment sensor and the presenting molecule sensor. The second situation represents a tissue transplant from a sibling. The protein fragment here generates a response from the immune system. Any such small molecule which is recognized and causes the immune system to mount a response is called an antigen. All normal cells within the body which have a nucleus and produce proteins are capable of presenting antigens to the immune system. Consequently, all of these body cells are antigen-presenting cells. However, in order to present these antigens, they must be infected and eventually be killed. There are other cells within the body which are capable of engulfing entire viruses and bacteria, digesting them completely, and presenting these fragments on their surface without being infected. These cells are called professional antigen-presenting cells. We will discuss these in a later lecture. Normal cells within the body are called non-professional antigen-presenting cells. In this second situation, if we don't want the transplant to be rejected, we administer immunosuppressant drugs like cyclosporin to the patient. These drugs turn down the immune system. In the last situation, there's either an infection or cancer. Here we have a virus, viral proteins, and viral protein fragments. Bacteria, bacterial proteins, and bacterial protein fragments. A cancer mutation, cancer proteins, and cancer protein fragments. Here we have the virus leading to its fragment, the bacterium leading to its fragment, and the cancer mutation leading to its fragment. The last item to identify is the specific type of immune cell. The immune cell here is a special type of lymphocyte, a T lymphocyte, specifically a cytotoxic T lymphocyte. It is abbreviated T subscript C. This lymphocyte kills bad cells. And this is our special immune cell. So this concludes our lecture on immune system recognition using the major histocompatibility complex class 1 or type 1, abbreviated as MHC1. I hope you've learned a lot.